Right. Okay, everyone. Thank you so much for joining us today for Hatfield Marine Science Center's research seminar. My name is Cinnamon Moffett, and I'm the research manager at Oregon State University's Hatfield Marine Science Center in Newport, Oregon. I will be your host for today. Just a few little logistics. You've seen me work through that as you all have been logging in, but we do have your mics, camera, and screen shares turned off. If you can help us by keeping it that way for the duration of this talk, that would be great. We do ask that you interact with us with lots and lots of questions. We'll put John on the spot as much as we can. Just enter those questions into the chat box. You can find that chat box if you are not zoomed out and you haven't participated as many Zooms as I have in the last few weeks. You can find that chat by going to the bottom of your Zoom screen or the top of your Zoom screen. Click on the word called chat and you'll get a little pop-up box that you can put in the questions. Feel free to put in the questions at any time, but we'll get to them at the very end. I also wanted to let folks know that we are recording today's session. So if you missed something or you want to share today's session, it will go up on the HMSC's past seminar page in about 2 days. So you'll be able to find it there. I also want to put in a little plug for next week's seminar. We were doing a little bit of marine mammal kind of go get them here this month, but our next seminar, it will be on January 28th. It'll be Robin Baird from Cascadia Research Collective, and he'll be talking about the science management of endangered false killer whales around the main Hawaiian Islands. So excited to have another mermammal talk next week, so hopefully you can join us there. And again, if you are interested in learning more about any of our upcoming events, you can use whatever platform you use and enter HMSC, Google or whatever, and come to our homepage. You scroll to the bottom and you'll find a calendar of events. All the login information and links are there. So hopefully we'll see you again next week. But for today, today's speaker has been invited by Lisa Balance, Director of the Marine Mammal Institute at Hatfield Marine Science Center. So I'm going to hand it off to Lisa to introduce today's speaker.
Thank you, Cinnamon. Good afternoon, everyone. And I appreciate this opportunity to introduce today's seminar speaker. As Cinnamon mentioned, my name is Lisa Balance. I'm the Director of the Marine Mammal Institute here at Hatfield. I've been here almost 1.5 years now and beginning to get my feet underneath me with respect to all things Hatfield and OSU. And in the context of this speaker today, I must admit that I feel a lot more comfortable because I have a long and deep history with Dr. John Durban. He and I have worked together in various ways, shapes, and forms for well over a decade. And it's a real pleasure to have him here. I can tell you all we are in for a treat with this seminar. Dr. Durban is presently Senior Scientist with North Gulf Oceanic Society, Senior Scientist with South Hall Environmental Associates, and Associate Professor through a courtesy appointment with OSU's Department of Fisheries and Wildlife. Prior to these positions, he was leader of the Cetacean Health and Life History Program at NOAA's Southwest Fisheries Science Center in La Jolla, California. And that's where he and I worked closely together for 10 years. John has a Bachelor of Science and doctorate degrees from the University of Aberdeen. And his disciplinary expertise pertains to many topics, but especially population ecology, particularly quantitative analyses of abundance, demographics, and life history using photographic mark recapture methods, spatial assessments of movements and habitat use using satellite telemetry, and pioneering the application of Bayesian statistical methods. John has a long history of using photogrammetric methods to characterize condition as a measure of health, and the goal of that science includes mitigating threats before they can cause population declines. He's applied these methods to free-ranging cetaceans in the Pacific, in the Atlantic, and in the Southern Oceans. John is notably accomplished in terms of publications. To date, he has 90 papers in the peer-reviewed literature. He's also notably accomplished in terms of obtaining research support, with over $3.5 million obtained in research grants to date. So with that brief introduction, let me turn it over to Dr. John Durban to present Survival of the Fattest, Integrating Photogrammetry Methods of Health into Population Assessments for Killer Whales. Thanks, John.
Great. Thanks, Lisa. Can I just check that you guys can hear me?
Yes, we can hear just fine.
Okay, perfect. Well, I think you can see my screen. Yeah, thanks very much for the introduction and the opportunity to give this talk. It's always a pleasure to talk about killer whales, and so I hope this will stimulate some questions, and there'll be time for this afterwards. As you'll see in the next few minutes, this work is work I've undertaken with a number of colleagues for some time, for many years, but it was certainly advanced over the last decade as I worked with NOAA's Southwest Fisheries Science Center. So I wanted to acknowledge that on the title slide. And it continues to advance through close collaboration with Sea Life Response, Rehabilitation and Research, a nonprofit organization based here in Seattle. And so I'll try and give credit where it's due, but there's plenty of folks that have been involved in this work, and that continues today. Let me see if this is okay. So this is a team effort, and the best way I could kind of acknowledge that is just to outline 3 key papers that are currently in the late stage of review that are going to form the basis of what I'm going to talk about today. They're listed here, and I'll go through them more in detail, but one of those is led by Josh Stewart, my former postdoc at the Southwest Fisheries Science Center, who's been working with us on some of the analysis of southern resident killer whale photogrammetry data. So Josh and a team of folks have been underpinning this great work. Similarly, I'll be talking about killer whales in Antarctica, where I'm leading a paper looking at size and body condition, and my colleague Holly Fernbach is leading a paper currently on abundance estimates that I'll weave into this talk. But all the folks listed on this slide, and many more that I'll acknowledge have been central to this work. Particularly here, this is a photograph of myself and Holly. All the photogrammetry work I'm going to be talking about today has been both from manned aircraft and from our drone work has been accomplished by working together with Holly as myself and her as a flight team. So this certainly couldn't have been done without her assistance and great expertise. So going on to killer whales, there is a long history of interest in killer whales as top predators in marine ecosystems. In the 1st century, Pliny the Elder had this great quote that certainly will inspire young boys to get interested in killer whales, describing them as nothing but a mass of enormous flesh armed with savage teeth. And I think we've come on some way from that in our understanding, but it is certainly true they're apex predators. Here's a picture on the left of a southern resident killer whale eating a Chinook salmon, and on the right, a transient killer whale flossing with a harbor seal. And recent work over the last 20 years has really highlighted how, because of their large body size and high metabolic rates, they are large consumers, large top consumers in ecosystems, and they have the ability to affect top-down forcing on those systems. The concept I'm talking about today is really the reflection of that, in that as apex predators, they're also impacted by changes in trophic dynamics in the ecosystems that support them. And so therefore, studies of killer whales as top predators can tell us something not only about their health, but the health of these systems. So that's really the overarching concept that these case studies, I want to discuss them in. So where are we now? Here's the East and North Pacific if you're on the water, or the Pacific Northwest if you're on the land. The bottom arrow here is you guys in Newport. The top arrow is where I am. I live on Stewart Island in the San Juan Islands, about 300 miles up the coast from you. Particularly, this green building here is where I'm sitting now. This is my house. These are some transient killer whales in front of us. I've been conducting research here for the last 28 years. Over the last 2 years, I had a large career change and left my position at NOAA, mostly to be on the ground here and focus more on the science that I believe is integral to support management of endangered southern resident killer whales in the Pacific Northwest. This is an aerial picture of southern resident killer whales. They are salmon specialists. They eat predominantly Chinook salmon. They aggregate around southern Vancouver Island, where I live, particularly in the summer months, but they're here all year round, to feed on returning runs of Chinook salmon. There's a Chinook salmon in the picture here with part of LPOD pursuing it. There's been a checkered history between humans and killer whales in this part of the world. Killer whales have been revered by First Nations, and still are, But white settlers initially saw them as a threat, a competition with salmon stocks, and they were shot with Gatling guns and harpoons. More recently, for this population in the 1960s and 1970s, a few dozen were removed in live capture fisheries, particularly for display in aquaria. And one of these whales from the southern resident killer whale population, Lolita or Tokite, is still alive in the Miami Seaquarium today, the last one left in captivity. In the early 1970s, as marine mammal protection became enacted both in Canada and the US, there was renewed interest to look at the effect of these removals on populations of killer whales around Vancouver Island. And this started long-term studies, initially by Fisheries and Oceans Canada, by Mike Bigg and Graham Ellis on the Canadian side, but also in the early 1970s, my colleague and close friend Ken Balcom was contracted by National Marine Fisheries Service on the US side of the line to look at population censuses of killer whales in this area. Together, they pioneered what has now become a stock tool for us all of us studying cetaceans, which is called photo identification. We can tell individuals apart by taking pictures of their dorsal fins and saddle patch pigmentation, much like a human fingerprint. And in the case of the southern resident population, this has allowed every animal in the population to be documented, enumerated, counted, and their associations, social affiliations, life histories to be documented since the 1970s. Really an incredible data set that has allowed the kind of studies I do to be built on that. Believe it or not, this is a photograph of me 28 years ago, beginning my career at the Center for Whale Research, just on Salmon Island, near to where I am now, where I learned the value of studying individuals and long-term studies through photographs. At that point, in the early 1990s, southern resident killer whales had been increasing since that exploitation through the live capture fishery, and an increase from about 70 to close to 100 whales. Everything was looking pretty good. But in that decade, particularly in the 1990s, and following on to today, there's been a decline in abundance, and we now only have 74 animals in the population, the lowest abundance in almost 30 years. In early 2000s, they were listed as an endangered species by merit of being a distinct population, a distinct population segment, and endangered both in the US and Canada. In our early understanding of why this endangered status has come into effect came from a seminal paper led by my colleague John Ford, where he used these photo identification documentations to estimate southern resident killer whale mortality, and related that to coastwide abundance of their prey, Chinook salmon, and showed that there was a large decline in Chinook salmon abundance in the 1990s that coincided with an increase in the mortality of the southern resident killer whales. This really opened our eyes to the degree to which they're prey limited. Now, this integral relationship between salmon and killer whales has been known for a long time by the First Nations. I love this illustration on the right that shows a killer whale with a salmon in its stomach. But for us, it was only really in the last couple of decades that we've begun to understand this issue. Limited prey availability, and particularly access to Chinook salmon, is recognized as a key threat to this population in recovery plans, both in the US and Canada. And this has led to questions of, are the killer whales starving? Is this something that's leading to their trajectory to extinction? And how can we manage this? How can we mitigate it to maintain these icons of the Pacific Northwest? And this is really where my recent work has focused on developing methods and generating data that can address this. And particularly, can we identify times of food limitation so that we can do something about it before whales die? And that's really a concept I'll keep coming back to. So this initially started in the early 2000s, where I extended our photo identification methods to something called photogrammetry, which is simply making measurements from photographs. And we started off with what I call laser metrics, where we mounted two laser pointers on a camera. These laser pointers are a fixed distance apart, in this case 10 centimeters. We project them onto the dorsal fin of a killer whale. Therefore, we have a scale of known size. So we could do things like measure the dorsal fin growth of males as they sprout into larger sizes. So here's a plot from one of our early papers that shows the dorsal fin height of an adult female on the left, only about 60 centimeters or so, a range of males as they grow and go through a process that we call sprouting as their fins become large, and to the size of an adult male on the right. So we had some ability to monitor growth, certainly of some body appendages. But what we really wanted to do was monitor body size and ultimately body condition to have sensitive metrics of how the whales were doing. And our first chance of doing this came along in 2008, paper published in 2011, where with funding from NOAA's West Coast Regional Office, we were able to charter a helicopter and do what's called aerial photogrammetry, where my colleague Wayne Perriman at the NOAA Southwest Fisheries Science Center has pioneered this approach. But if we can take photographs from directly above the whales with a relatively long lens, we can have an undistorted view of size and shape. And if we know altitude and focal length of the lens through this ground resolved distance calculation below, we can scale pictures or measurements of pictures in pixels up to measurements of real size. And so this is something we were able to do in 2008, first of all, for southern residents. One of the really key things with this study is we were able to link images taken from several hundred feet in the air to known individuals with known life histories through these long-term photo identification catalogs, because we could see their saddle patch pigmentation. So we can see that this here for one individual, a young male in K-pod. You can see the bottom shows the boat base identification photographs. He's got this wonderful asymmetrical saddle pigmentation behind his fin. And the top shows images taken from an aerial perspective where you can clearly see those same markings. So we could link our measurements, particularly to known age and sex. So as part of her PhD thesis work, that Holly was able to come up with the first size and age relationships for this population. And this is one of the key figures. And it shows that males in gray were growing to about 7 meters in length. Females in black about 1 meter shorter, about 6 meters in length. And notice at this point, we had animals up to close to 60 years old, confirmed ages. One of the key things we found, however, was that growth was not uniform to an asymptote. In other words, adults, particularly the adult females that were older adults, had grown to larger body sizes than young females of adult age. These young females, they typically mature by the age of 15 or so, but they were growing to a length on average only about 30 centimeters shorter than the old females were. And particularly, these shortened animals were the ones that were growing up during the period of low Chinook salmon in the 1990s. We believe it showed an effect of not only did this shortage of Chinook salmon affect mortality, but it affected the condition and growth of whales that lived through this. And that was our hypothesis. But one of the problems with studying endangered species is your sample size is fairly limited. For example, see the adult males here. We didn't have very many old adult males to look at that pattern. So what we did is take the same approach and try and look at the neighboring population of northern resident killer whales that aggregate around the northern end of Vancouver Island. This plot here, not quite up to date, but it shows that during the course of the photo ID study for southern residents in blue, northern residents have been at much higher abundance and have been growing in abundance. But if you look at the gray bar, during that period of population decline for southern residents in the late 1990s, northern residents also declined. So they're somewhat correlated, and it gives us a replicate population to look and see if the same effect of body size also occurred. So we've collected aerial data from these two areas on the bottom right here around southern Vancouver Island with southern resident killer whales, and the top left around northern, with northern resident killer whales around northern Vancouver Island. This is work we've done with my great friend Lance Barrett-Leonard from the Vancouver Aquarium. And lo and behold, in a paper we just published last year, we saw a very, very similar pattern. This is not the southern residents you're looking at, this is northern residents, and we find a very similar pattern, particularly these animals. I just highlight the females here, again, larger sample size. The old females, in this case over 40 years old, were growing to almost 0.5 meter longer on average than the younger females that from 20 to 40 years old. So just putting them side by side, here we see the pattern for southern residents and northern residents, very, very similar. Interestingly, this isn't a function of continued somatic growth over time. These older females, the larger females were over 40 years old in northern residents and over 30 years old in southern residents, simply because these studies were conducted 10 years apart. So in other words, they weren't changing growth at the same age, they were changing at the same time. These younger animals growing up in that period of Chinook salmon decline were smaller. So this was strong evidence that there was a sublethal effect on individual condition and growth in this distinct period of low prey availability. Let me just check in and make sure you guys are all hearing me before I move on.
Sound
fine.
Okay, great. So when we started the study on southern resident killer whales, we're in a fairly accessible environment here. We're close to Victoria and Seattle. We had access to helicopter support. But when we wanted to replicate this work for northern residents, we were going to a much more remote location. And to collect aerial data, to fly a camera over whales, we pioneered the use of drones to collect aerial photogrammetry data from whales. In 2014, we conducted the first flights that we're continuing today. For us, drones are simply a method to get a camera above the whales. They're a flying camera. They've subsequently proliferated, of course, in their use in many fields of biology, certainly in studies of whales. And they've shown to be cost-effective, safe, and produce higher quality data than the manned aircraft for many missions, including counting, behavioral monitoring, and certainly for photogrammetry. What I like about it is that it's non-invasive science. Here's a picture of one of our smaller drones above a northern resident killer whale. It's about 130 feet above the whale. The boat where I'm flying from is several hundred feet away from the whale as well. So we're able to basically give them a health check without them even knowing we're there. I think that's a wonderful aspect, particularly for endangered populations. The other thing that I don't think any of us were really prepared for when we started this is we would get much higher resolution images, simply because we're flying effectively the same cameras much lower than we were able to previously. With manned aircraft, we were, by permit restrictions and to avoid disturbance, we flew several hundred feet above protected marine mammals. With drones, we can fly much lower, possibly as low as 80 feet or so without disturbing them. So that was really effective for us. By photographing standard resolution targets, we're able to look at the resolution, i.e. the distance that we can resolve in 1 pixel. Initially, when we started out at about 100 feet or 30 meters, each pixel was only about 1.4 centimeters of distance. So we could look at really subtle changes in shape of whales. This is what it looks like in the level of a whale. A photograph taken from about 100 feet, and you can see we can clearly make out differences in shapes and lengths and just absolutely spectacular images. Since then, we've graduated to using bigger drones. Unlike many aspects of technology that get smaller, for us there's a value in larger for a number of reasons. One, we can fly from much further away and still see the aircraft. So the picture on the right shows one of our large octocopters. It's 42 inches across. That's Holly holding it on the left. Because it's large, I can see it from further away. We can fly over whales from several hundred yards away and not disturb them. Also, because it's large, it uses large motors and propellers spinning relatively slowly, so it's whisper quiet compared to some of these irritating drones that folks buy off the shelf. And also, by being large, it has high payload capacity, so it can carry heavier and bigger cameras. So we're now able to carry full frame, much higher resolution cameras than we started using 6 or 7 years ago. And these are the kind of pictures we obtain. This picture just taken a couple of months ago, just around the corner here of K-Pod, one of the aggregations in the southern residents. And you can see how spectacular these kind of images are in resolution. It gives us great resolution for taking measurements, and that's what we want to do. So now we're really getting into this, being able to have the tools to address the issue of, are the killer whales starving? Because now we can look at body condition, not just length. So what we've been able to look at images of whales that have subsequently died and develop sensitive indices of body condition that we can measure. And particularly, we focus on an area around the head region. This photograph shows 3 whales, one on the left that's still alive, one in the middle that died about a month after imagery, and one on the right that died about a week after this image. And you can, if you look at the eye patches on their heads, you can see some very clear differences. The whale that's alive, those eye patches get wider apart as you move down the body. It's got this fat reserve behind the cranium. As cetaceans, particularly odontocete cetaceans, get nutritionally stressed, they metabolize that adipose tissue faster than they do some of their blubber, which leads the eye patches in killer whales to trace the shape of the skull. And that's what we see clearly in these animals on the right. So we looked at this and decided we can make some really sensitive measures of body condition by taking measures of this eye patch divergence or even convergence around the shape of the skull. So we developed an index of body condition called the eye patch ratio, which is simply a ratio of the distance between the eye patches at the front of the eye patches compared to 75% caudally along the body, which corresponds to behind the skull. So in other words, we're saying, are the eye patches tracing the shape of the skull or not? This has proven to be very sensitive. It's very repeatable. To demonstrate this, these are repeat photographs of the same individual southern resident killer whales in September, later in May when she's gone through the winter, and then in the following September. And we can see that in the September time, look at her eye patches. She's much more robust. There's much more fat behind the skull, but she's very lean in May. In fact, those eye patches are almost converging behind the skull. To look at that, what this relates to in data, this is that same female, the time series a bit expanded here, but this shows how she's fatter in the fall than she is in the spring after going through winter. The error bars here show the variability in that measurement with repeat measurements in the same season. And you can see that small compared to the effect size of changes between seasons. So we've got a very sensitive index. There's small error compared to the effect size. That means it's very powerful. Since we started our photogrammetry studies in some 13 years ago, we've been able to generate time series of this condition measure, this eye patch ratio, for each animal in the southern resident population. And that's what these gray horizontal lines are. They are the time series of measurements for each individual. What I've got in black here are the measurements for individuals that have died during the course of our study. And what I want to draw your attention to is particularly those individuals since 2015, when we've had repeatable funding for annual sampling. During that time, of those 10 deaths, 8 of them have clearly shown reductions in body condition prior to death. So to us, this was depressing, of course, but also exciting because it shows that we were able to document terminal declines in condition, but to do it before the whales died. So this is starting to offer the potential to generate data that could support management before whales die. Now, to formalize this a bit more and look at the relationship between body condition and mortality, we partnered with Josh Stewart, who was my postdoc at the Southwest Center, still a very close collaborator. And he has a paper in review where he developed a population model for this linkage. And there's a lot of information on this slide, so I'll step through it. But effectively what we've done is look at, for each eye patch ratio measure we make of each whale and each year, we want to compare it to an expectation. Now, we expect the body condition of these whales will change somewhat through their lives. And that's what these lines are, the blue for males and the red for females. These represent a generalized additive model fit to all the measurements we've taken. And they show, for example, that calves and juveniles are a little leaner, a little skinnier than adults, simply, particularly for these juveniles. We call that a juvenile slump. Because these juveniles are learning to feed on their own, they're growing, there's energy given to growth, not just maintaining body condition. But so we have by having data over the course of 13 years, we can generate these expectations. Then for any given measurement that we make, we can look at the residual, where the distance of each of those points from the model line, and generate a distribution of those residuals. So that's what we do. And then we divide that distribution into 5 20 percentile classes. So the lowest 20 percentile will be animals with a negative residual, i.e. they're in poorer condition than expectation. The upper 20 percentile will be animals with a positive residual that shows them fatter than expectation. So by doing that, in any given year, we can place an animal into a body condition class, one of 5 classes. And to visualize what this looks like for a whale, on the bottom, this is J17, an adult female that passed away a couple of years ago. And on the plot at the top, her measurements for 4 consecutive years are shown by the black circles and the black lines. And so in 2015, you look at the image there, her eye patches diverged as you went caudally past the cranium. She was in great shape. She was in body condition 5. But by the time her last year, we saw her in 2018, she had declined in condition to body class 1. So you can see this decline in condition before death. What Josh is able to do is look at a multi-state model to look at the probability of state transitions between, say, body condition 1 and 2 or 4 and 5, and model these in terms of parameters for whether they stay stable, growth, or decline in condition. these state transitions allow us to model the probability of state transition in terms of covariates. And that's something that Josh is currently doing, particularly looking at the link of body condition to salmon abundance. And I'm not going to cover that here, but it offers great utility. What I wanted to demonstrate here is we can also look at the probability of transitioning from any body condition class to death, to not being seen the next year. And this is our way of formalizing a link between body condition and mortality. And of course, before we do this, we have to control for variable mortality across age and sex classes. The top plot shows this. The base mortality probability is adjusted for different age and sex classes that the whales are in. Clearly, there's a higher mortality probability for example, old females and old males. But once we've controlled for this, the bottom plot then shows the effect of body condition class. And what we clearly see is that body condition class 1, there's animals in the lower 20th percentile of the residuals under the line, so they're thinner than expected. They have a much higher mortality probability for the subsequent year than any of the other body condition classes, 2 to 3 times higher. So this has really formalized our view that our body condition is a predictor of subsequent mortality in the coming year, and therefore can potentially provide an early warning system. And I'll come on to that in a moment. What that looks like in terms of relationship to the population numbers, the southern resident killer whale population is made up of 3 large social aggregations: J-pod, K-pod, and L-pod. They have slightly different dynamics, so I split them apart here. But what this plot just shows is the count, the photographic census of each pod in black over the time of our photogrammetry time series. And in red is a condition index, which is simply the proportion of the individuals in body condition classes 2, 3, 4, and 5, i.e. not in the poor body condition class. And we can see there's a tight correlation. Typically when body condition starts to decline, abundance declines, often with a 1-year time lag, as Josh's model has shown. So mortality, body condition is predictive of mortality. So I believe this provides strong evidence that we have an early warning system. And I want to go back to J17 here, this female that died. The plot on the bottom, the black shows her body condition index over time. And you can see that since 2015, where she was fat, pregnant in the image above, she's declined steadily and quite dramatically in body condition. But we've been able to document this clearly every year. So we've been in a position to say, hey, J17 is declining. And so I think that this offers some tools that as she moves into this body condition class 1, there should be some warning flags developed and hopefully some enhanced management actions taken. And in the last couple of years, we've started to see this coming into effect, which is really exciting and encouraging. For example, some of our images of whales in poor condition and whales in late stages of pregnancy are now being used in what I would term enhanced management responses. For example, calling on users on the water, whale watchers, fishermen, to give the whales more space, to provide acoustically quiet areas to increase their foraging success. I think that's something that's incredibly valuable at a kind of community level. And just the last couple of weeks, you may have seen that the Pacific Fisheries Management Council has agreed to start developing adaptive management frameworks to change fisheries harvest policies depending on both the number of fish available out there and also the condition of and abundance of the killer whales. So we're now starting to work with our colleagues in NOAA to develop adaptive management procedures that would use these kinds of data, again, to help try and provide more food for the whales before they die. So I think this is a long-term study. We're by no means finished with our tool development and application, but I think we're starting to see some exciting avenues that hopefully aren't too late for the whales. I'm going to move on to Antarctic killer whales now. Can I just check that you can still hear me okay?
Sound good, look good.
Great, okay. So here I want to now turn to killer whales in Antarctica, which is the most spectacular place on the planet to study killer whales. Probably over half the world's killer whales live in Antarctic waters, or at least occur regularly there. These are special killer whales. I want to say a big thank you to Bob Pittman, who I know is in the audience today and is a new member of your community down in Newport. Bob gave me the opportunity 12 years ago to join him on his Antarctic killer whale research. This is a photograph of Bob and I on my first trip to Antarctica with the BBC Natural History Film Unit filming Frozen Planet. And it's really been one of the great joys of my career to work with Bob, and I've learned a lot, and much of the data we've collected here has been inspired by his interests. So thanks, Bob. Over 100 years ago, early explorers described being attacked by killer whales on the ice in Antarctica. But it took almost 100 years for us to really start understanding or learning anything about killer whales in Antarctica. And thanks to Bob's work and work that we've been able to do in collaboration and following up from that, we've now identified 5 distinct ecotypes of killer whales in Antarctic waters. These differ in how they look, in their pigmentation, in their size, as I'll show you. They differ in their ecological role. They eat different things. They're genetically dissimilar. They perhaps have species-level genetic differences. It's a remarkable assemblage to study and understand. I'm going to focus on the 3 on the left here, because these are 3 that occur in the coastal waters around the Antarctic Peninsula. As many of you may know, the Antarctic Peninsula is one of the most rapidly warming areas on the planet. This is depicted by this plot of temperature anomalies for last year. This is leading to a dramatic decrease in sea ice. You can see on the west side of the peninsula here, there's very little sea ice, as shown in white. This is the part of Antarctica that sticks up furthest north, is pointing up towards the bottom end of South America. So there's an intense interest in understanding the effect of these environmental changes on the ecosystem there. And killer whales as top predators, I believe, give us a window into changes in trophic dynamics and ecosystem function and structure beneath them. These 3 types of killer whales, I've just stepped through them really quickly. They're dramatically different. One type we call, or Bob named, type A's, which is a large black and white form, looks very like killer whales elsewhere in the world, but they're probably the largest killer whales on the planet. They hunt in groups and consume large whales, particularly Antarctic minke whales. Here's a killer whale taking an Antarctic minke whale, one of the animals we documented on that first filming trip with the BBC. They also eat elephant seals and some other pinnipeds, but Antarctic minke is a big part of their diet. Type B1 killer whale, these are, I think, are the most dramatic animals, spectacular animals on the planet. You can see they look very, very different to other killer whales. They've got this cape patterning, a huge white postocular eye patch, much larger than a regular eye patch. And they occur in a really odd place for an air-breathing dolphin. They occur deep in the pack ice because they're looking for seals like this Weddell seal. We call them pack ice killer whales. And these are the type of killer whales that have now become famous for this extraordinary show of cooperative behavior, of creating waves to wash seals off ice floes that we're lucky enough to have seen dozens of times now. It's just the most spectacular behavior I've ever seen. And then the third type that over the last 10 years we've defined and described, which is, we call them type B2 killer whales. They have a very similar pigmentation pattern to those B1s. You can see from this aerial image, they've got light lateral sides, a dark dorsal cape, and very large eye patches. But they're much smaller animals. They're much more gregarious. They occur in large groups. They're vocally active. They're not hunting with stealth like the B1s are. We see them eat penguins, but we also know from our work with satellite telemetry, with telemetry-linked dive tags, that they regularly take deep dives down several hundred meters, up to 700 meters, where they're likely feeding regularly on fish and squid. Our work, largely over the last decade or so, is focused on using this stock tool of photo identification to recognize individuals. Unlike the southern residents that occur in small population sizes in a relatively small area where we can find them, we can't count Antarctic killer whales, or at least not yet. We have to sample them, in other words, take photographic samples and estimate abundance using photographic mark recapture techniques. These are the kind of photographs we get. There's some B2 killer whales, 2 different individuals. On the top, you can see one individual photographed in different years. It looks slightly different. In one year, it's got a heavy diatom coating. It's been in cold Antarctic waters for a long time and stopped sloughing its skin. On the bottom, there's another individual that you can see we can just tell it apart from the top one based on its dorsal fin profile, but also on its distinct saddle patch pigmentation. So we collect these kind of data. We generate capture histories for the individuals we see, when we see them, how often we see them, and we use statistical models to estimate how many we're missing. I'm not going to go into those models in detail today. That would be a whole different talk that I hope I have an opportunity to give at some point. There's some exciting challenges there. But one of the key tenets of mark recapture is we have to define what our populations are. And we're lucky for all three types of Antarctic killer whale around the Antarctic Peninsula. We have quite a high site fidelity. We know this through our satellite telemetry, but also through these plots show the dots are locations where we've collected photographs from different groups, and the lines are matches of individuals between areas. We have a lot of site fidelity between years. Although these animals may travel quite widely, in the summertime they come back and feed in the same areas. And this gives us an opportunity to define that we're estimating the abundance of these types in the coastal waters on the shelf on the west side of the Antarctic Peninsula. Recently, in two papers, we've been using these photographic mark recapture techniques to estimate abundance of the three types and estimate trends. What you see here, the dots are annual abundance estimates. The dotted line is a kind of model trend, and the gray is the 95% probability intervals from a Bayesian estimate of abundance. And these are what I want to draw your attention to first. Because we're not counting these individuals, we by no means see them all every year. We're estimating abundance, and so there's uncertainty associated with those estimates. That uncertainty can be great in years where we don't have a big sample size and we have a low capture probability. It's also great when the population is really high. And if you look down at the B2s here, at the bottom right, you'll notice that scale is going from 0 to 2,000 whales. They occur in much higher abundance, on average about an order of magnitude higher than the type A's and B1s. So those mammal eaters, the A's and B1s, are occurring in abundance of about 100 each. But the B2s, on average, an annual population estimate is close to 1,000. So because we have lower capture probability on any one of those individuals, there's a lot more uncertainty. This uncertainty has two effects. One, it means that we can be less sure of the trend that exists. And what I've imposed on each of these plots is the probability that we think a trend exists. But it also means that we need long time series to detect significant trends. This is work that Tim Gerardet at the Southwest Center did years ago that really did power analysis and introduced the idea that it does take long time series when we do sample-based abundance estimates. So we need another method of looking at current assessment, and that's where I'm going to come on to photogrammetry. But I want to draw your attention to what we do know about contrasting trends here. Type A killer whales in the top right. Remember, these are animals that like open water. They feed on minke whales. They're increasing in abundance, and we have a high probability that they're increasing, over 90% chance. And they're increasing and apparently doing well from increased access to ice-free waters as we see more ice melt, and also increases in minke whale abundance, elephant seal abundance in those ice-free waters. In contrast, the two types that are pegophilic or ice-affiliated, the B1s particularly, those that like the pack ice killer whales, they're decreasing in abundance, again, with a fairly high probability, fairly high likelihood. The B2s are likely around stable, but again, because of this uncertainty, we really can't tell exactly where they are on their population trajectory, but what we do know is they're in very high abundance. So to try and understand, have a bit more sensitivity to what's currently happening, how are they responding to an environment that's changing so quickly, we've recently, or over the last 5 years now, taken our photogrammetry techniques with drones to Antarctica, which has been remarkably successful. You can see from this image, it is a stunning place to conduct field work. If you look at the little dot, our drone over 100 feet above this type B2 killer whale, and that's us in the small boat several hundred yards away. Again, non-invasive science. With these killer whales, we don't have 40 years of data like with southern residents to relate photogrammetry measurements to known age, but we can look at age classes. From our aerial images that you can see here that are just spectacular, we can identify adult males, for example, the animal on the left, because they have these much larger pectoral flippers and also curved tail flukes. And similarly, we can identify adult females like the one on the right because of the presence of a dependent calf that often travels in this echelon position. So because of that, we can at least compare sizes and body condition of whales in different sizes, but also in these states of being an adult male or an adult female. And so in a paper that I have in review currently, we've been able to now scale these three different types around the Antarctic Peninsula into real size. And the illustration on the left now is scale for length. Those are wonderful illustrations by Uka Gorta, who's based here in Seattle. The plots show frequency plots of our size estimates for each of the three types. And the black dots on the x-axis show the sizes for adult males, which are typically non-overlapping and larger than the sizes for adult females, which are in the gray dots. And you'll see from this that the type A killer whales, which I previously said are probably the largest on the planet, can range up to 9 meters in size and massive. The type B1 killer whales are similarly large. The animals almost up to 8 meters in size. But the B2 killer whales, the most abundant form, are diminutive. They're much smaller, typically only 6 meters or so. Now, why does this matter? It matters massively if we're trying to think about their contribution in terms of energetics as top predators. How much do they eat? How much do they need to eat? For example, an adult male, we can extrapolate from length to mass based on relationships from the live capture fisheries, ironically, here in the Pacific Northwest in the 60s and 70s. They're not going to be ideal for Antarctic killer whales, but it still shows that if you add length for a killer whale, you add mass exponentially. So, even a few centimeters of additional length gives you a huge amount of extra mass. So, for example, the average male type A killer whale we've measured is 7.6 meters long, and that would translate to a hypothesized weight of 5,500 kilograms. In contrast, the average male type B2 killer whale is over a meter shorter, but that translates to a difference in almost 2,000 kilograms, over 5,000 pounds weight difference between adult males of these different types. Massive differences in sizes, and I think this data will be really useful as we move forward to parameterizing ecosystem models. But perhaps most interestingly, what we found is differences in body condition between the types. These three just graphically show a difference that's quite consistent, where type A killer whales are long, but not necessarily very robust, compared to the type B1 killer whales, which are just massively robust whales in general. And the type B2 killer whales, again, are not only diminutive in length, we often find them in quite skinny condition. If you look at the eye patches on the whale on the right, you'll again see that they're tracing the shape of the skull on that whale. Now, to index body condition here, we clearly can't use the same eye patch ratio because the eye patch characteristics of these three types vary so much. The B1s and B2s have long eye patches, much larger than the A's. So what we've done to compare them is just take a measure behind the skull, a measure of head width at a fixed distance behind the skull. And in this case, it's 15% of the distance between the blowhole and the anterior insertion of the dorsal fin. So we can compare types. And again, because we don't know age, what we've been able to do is relate this to total length measurements. So here we have a plot of the body condition in terms of head width for the three types regressed on age. The blue line is type A's, the yellow line here, the B1s, the two large forms. You can see they largely have the same distribution of length estimates, kind of the same body condition distribution, although B1s, you see that line is more steeply sloping. Large B1s get more robust, significantly more robust than type A's. But the diminutive type B2s are noticeably skinnier. If you look at that gray line, it doesn't overlap really. It doesn't even overlap with the error bars of the other two lines. It's significantly different. The equations on here show the regression equation if we center it on a total length of 6 meters. And I've done that so that we have overlapping length ranges between the three types. And if you look at that, the body condition, the head width of type B2s, even at the same length, is on average is 0.68 centimeters for a 6 meter whale compared to, 0.68 meters, compared to 0.78 meters, so 10 centimeters smaller than the other types. So, the type B2s are diminutive in length, but they're also much, much skinnier currently. The shaded dots on here show outliers. So these are animals that I've identified as being at least 2 standard deviations away from the fitted trend model. And you can particularly see that we've got a significant number of low outliers. Think of those low residuals for southern residents. These are animals in poor condition of type B2 killer whales. And these are largely in the last couple of years that I think is a sign of perhaps impending population issues. I'll just show you what these guys look like. Remember those emaciated southern residents? Well, these whales look even more emaciated. Look at the shape of the skull. It can clearly be seen. They've really got very little subcutaneous fat. These are two different females, both with calves, and you can see with the calf on the right, it too is an emaciated whale. So we've now seen several of these whales. They seem to be becoming more common that we're seeing these whales in poor condition. But even the type B1 whales, the really robust ones, we're starting to see whales in poor condition. If you look at the picture on the top left here, it's a group of 5 whales. Well, the adult female on the bottom, you can see is so skinny. Not only can you see the shape of her skull, you can see her rib cage. We're starting to see B1s in this kind of condition as well. The photograph on the bottom of the right comes from a paper we recently published with Rebecca Hooper looking at skin microbiome, but it shows that in addition to whales that we're photographing, other people are seeing B1s in really poor skin condition. Again, sign of poor body condition for sure. So, you know, through our abundance estimates, we're learning about general trends over the last decade or so in abundance, but we don't have much power to look at year-to-year variability. With photogrammetry, we're now starting to reveal a pattern that we might be seeing impending changes in body condition of these ice-affiliated forms that I believe, based on our work with southern residents, will result in us likely seeing declining populations in the coming years. I don't think it's any coincidence. These are the two types, the two pegophilic ice-affiliated forms of killer whales we see. They need ice to feed, particularly in the case of B1s. In the case of B2s, that ice certainly leads to productivity for what they're feeding on and for the types of penguins they like to eat. This is a picture that I took on my first trip to Antarctica with Bob of type B1 killer whales. And if you look at the animal on the left, that's just a massively fat whale. When it spy hops like that, you just see it rippling with fat. That's what a killer whale needs to look like in Antarctica to survive. And everything down there needs to be fat to survive the extreme cold. And it's really worrying. In the course of a decade, we're starting to see very different, very skinny killer whales in that environment. And so we'll be continuing this work, and I fully expect to start seeing abundance declines as well as declines in condition. I just wanted to start wrapping this up. This is a picture of a southern resident killer whale, and if you look really closely on the bottom right, you can see it's about to suck up the Chinook salmon. You know, I think it's a really graphic and beautiful illustration of this connectivity between top predators and the ecosystems that support them. I think that we've started to develop tools that really augment these long-term studies that I'm invested in and I believe in. We've got to continue this population monitoring, but there's ways we can augment them and learn about individual health, quality, not just quantity. And finally, this is a picture taken last night. We were out with southern residents late into the evening, and it really, to me, emphasizes long-term studies. This whale, J26, a male who's almost 30 years old. When I first started research here, he was only 2 years old. So I'm sure he's aged better than I have, but I think it just shows the value of these long-term studies in terms of what we can learn about the whales, and we're adapting our tools to speed up our inference to help conserve them. So I'll end there, but I'm happy to take any questions.
Thank you so much, John. It's fascinating work. For folks that are online, please put any questions you might have into the chat box so we can have John answer them. And while we're waiting, I just want to make a correction. When I introduced John, I said that this video would be available in a few days. It'll probably be available in a few weeks while we wait for John's publications to get finalized. But if you do need a copy of this, just go ahead and contact me and we'll be able to do some internal work. But if anyone has questions, go ahead and put them into the chat. And I have a question while we're waiting for folks to get started. John, have you seen or been able to document a tipping point in body condition on the southern where you anticipate they're not going to be able to come back no matter what kind of adaptive management is offered?
Yeah, well, I think that the work we've done with Josh showing that animals in that body condition class 1, so the lower 20th percentile of all the measurements ever made, they're the ones that have this high or elevated mortality risk. So almost certainly those animals like that, they haven't, some of them make it. I mean, you know, there's some variability there, but they're the ones that we consider what we call animals of concern, that we report to NIMS and DFO in our reports that we do based on our field work. But once we really start seeing the shape of the shape of the skull from those measurements, it's a display on what we call peanut head condition, where you can really see it. Those animals don't come back. Once they look like that, they just decline to death. And that's what before we had aerial photographs, that was really the first glimpse you got from a boat that animals in poor condition. From a boat, you can't see the subtle changes. You only see the really dramatic ones. And by the time you see that kind of syndrome where you can see the shape of the skull, those animals are almost certainly going to die. So by that point, they're beyond the tipping point. But based on these increased mortality estimates of animals in this lower 20th percentile, that's when they drop into that shape, that's when we really call them animals of concern and want to try and push and encourage, you know, kind of further follow-up and mitigation.
Okay, now we're getting our questions in. So the first question I see here is, what methods do you use to determine age of the individuals? Can you expand on that? You talked about it a little bit with the
Yep.
photo ID, but
Well, we're really lucky with Southern residents that studies on this population began, or photo ID studies began in 1974. So really, much of the population is known by direct aging. That you know, we observe them in the first year as calves of their mothers. We follow them through life, so we know age, you know, to within a few months. For those few animals left that were adults at the start of the study, they were aged in terms of you know, whether they, you know, we had a minimum age in terms of whether they had reached reproductive age at that time, or if they were post-reproductive senescence. So there are some old animals that we believe reached 100 years old. They're not alive anymore. But almost all the animals in the population now were either observed born or weren't mature. They were under 20 at the start of the study. So effectively, we've observed those ages. And again, with the Antarctic killer whales, we don't know ages, but we were able, based on physical characteristics, to identify who was adult or not, to provide those comparisons between types.
long-term studies, valuable.
Yeah, absolutely.
So can you talk a little bit about the consideration or techniques for using photogrammetry when you use a wide angle lens?
Yeah, so we use what are called normal lenses, which means that there's, well, effectively that's in reference to the dimensions of the diagonal of the frame. And that means that there's no distortion across the frame, so we can do any measuring. Typically, for a 35mm camera, that would be a 50mm lens. For other sensors like micro four thirds, you know, little mirrorless cameras, that, you know, they have a crop factor that could be as small as a 25mm lens, and it's still flat and normal. If you use a wider lens than that, you're going to have some distortion, which means that the center of the frame might be fine for taking measurements, you know, and by flat, in that case, I mean that a pixel is equal in dimension in all angles. But as you get to the edge of the frame, you get that distortion effect, like we see in cell phone pictures or GoPro video, where things get stretched out at the edge of the frames. So it's not optimal for doing photogrammetry, for sure. There are software out there that tries to correct for that, and you know, they probably do a very good job of that. However, you have to correct the images almost on a one-to-one, one-by-one basis. And in our case, I mean, for example, last night we collected 3,000 images in about 2 hours. Suddenly you have a lot of images to do. So our approach is to use a relatively long lens. The long lens has the other advantage that to fit a whale on the frame, you have to be higher. And again, I like being high because we don't want to disturb the whales. We don't want there to be a sound footprint that affects their behavior. So if you have a wide lens, to get a high resolution image, you're going to end up having to fly lower to get the whale fairly big in the image, and by being lower, then maybe just some disturbance. And so, you know, it's not the end of the world, but having a flat lens or a long lens is definitely preferable.
Nice. And Joshua, if that didn't answer your question, go ahead and type in additional question, and I'll ask that as well. So there's another question down here that I think you might have to explain a little bit, but are you aware of any continuation of the milk study that was started a few years ago? The only, did you say milk study? That is what is written here. Erica,
I know of one milk study. Our colleague Dawn Noren from NOAA's Northwest Fisheries Science Center was doing a study based on whales at SeaWorld that was looking at contaminant transfer between mothers and offspring through the milk. And fat transfer. So perhaps that's what's being referred to. I'm not sure. I don't think Dawn's currently doing that, but I know she's got some results she's writing up on that, which is very interesting. And you know, you have by being able to access those whales at SeaWorld, she was able to compare circulating contaminants in the blood compared to in the milk, for example, and those kind of things. So perhaps that's what the question refers to.
That is the study, according to Erica, that she was referring to.
Okay, yeah. So I believe that I know Dawn's got some fascinating results from that. I've seen her present on that, so I'm sure there'll be a paper out on that pretty soon.
Okay. So there's another question about the plot. Oh, it just moved. Hang on just a second. It's hard to keep up when they're coming in quickly. There's a plot of mortality probability versus body condition class that you showed for the publication and review that looked like a U-shaped trend. When I would have expected the linear decline in the B1s most likely to experience mortality versus the B5s less likely to experience mortality. Right. What do you think is the reason for this U-shape?
Yeah, that's a great catch. One of the problems is that there are actually very few animals in that B5 class. So there's a subject to some uncertainty, but also most of the animals in that class, there was a gap between when they were imaged and when they died of several years. So in other words, that condition class 5 didn't really reflect what condition they were in prior to death, unfortunately. And so that way it looks like there's a kind of positive skew to that distribution just by having a few whales in that situation. Whereas, there's another plot in Josh's paper that I don't have here that looks at the time to death of whales in various condition classes. And of course, by the time you're in a condition class 1, you're close to death. So those animals are typically imaged closer to death. So we have much more certainty about that effect. So that's all it is. It's unfortunately a sampling artifact and it's driven by those early years of the study where our first 2 sampling years are 2008 and 13, 5 years apart. It took us a while to get traction and funding to repeat this. And that led to a lot of uncertainty. So whales that were in condition class 5 a couple of years before, we don't know exactly what condition they're in before they die, but we do in more recent years for these whales in poor condition. So it's, when that paper comes out, I direct you to, there's another plot, which is, time of image to the time of death, which really explains that sampling artifact. But it's a good catch, absolutely.
So Lisa, let us know if that didn't answer your question, but I think that addresses what you had asked. And then there's a great, Lisa says yes, there's a great question here about what do you think are practical things that can be done for the southern resident population survival? Is it limited funding? Is it, what are your suggestions for
yeah, so it's difficult. So for those folks that don't know, there are 3 main risks that have been identified. One is limited prey availability, one is contaminants in the environment, and one is disturbance, acoustic disturbance, largely from vessels. Those things are compounded by limited prey availability. If there's not much prey, disturbance that affects foraging, becomes more of a problem. Similarly, if there's not much prey and they're metabolizing toxic blubber, that becomes more of a problem. So ultimately, the big issue that we have to deal with is giving them enough food now. And there's really 2 sides to that. One is a long-term goal of salmon abundance. And, boy, there's been a lot of money, billions of dollars thrown into that by NIMS in the last 30 years. We want to restore salmon, not just salmon for resident killer whales. So that part of it is a really hard nut to crack because many of these processes that are going on affecting salmon abundance are ocean-wide. It's climate change and warming trends that's affecting the proportion of salmon that come back. So that's really hard. But the more immediate one, these whales are on a trajectory to extinction more immediately. We need to increase accessibility to salmon, not just the ones that are out there. And that's where issues like keeping boats further away from them to avoid acoustic disturbance, giving them the best chance of increasing their foraging efficiency, I think are really important. And I think there's some good steps being made in these partnerships where we identify whales of concern and folks give them more space. We know that vessels of different kinds do reduce their foraging efficiency by masking, by acoustic disruption. So that's something we can do. I think ultimately, there's a lot of similarities between conservation efforts for southern resident killer whales and those of North Atlantic right whales in the Northeast. I work on right whales as well. We know the problem there. Those whales are getting entangled in fishing gear. We know the issue here. The whales aren't getting enough food to eat, in my opinion. I think these are societal decisions to be made. Do we decide to eat lobster from the Northeast? Do we decide, do we decide that whale conservation is more important? Do we eat sustainably caught Chinook here? Do we want to go out and watch whales closely on whale watch boats, or are we happy to watch them from shore? All these things I think are society's decisions. So there's, unfortunately, in the case of southern residents, we need to do things quickly. Well, in the case of right whales, but I don't think that there's a huge amount of effort going into that first problem, the salmon abundance issue. From restoration efforts to fisheries management. I think the big issue is giving them access to the food they need now to reverse the trajectory. So those are the things like managing vessels, area closures, fisheries where the whales are now, giving them access to the fish that are out there. But it's a really difficult issue. There's lots of other components to this that I haven't mentioned. For example, inbreeding is increasing in this small population. That might be leading to fitness issues we're just trying to discover. It's not all about food, but food is, animals need to eat.
Yeah, Definitely. There's one more question here that takes us back to the body condition question. And for folks that are online, I know that we're pushing our time limit. So thank you for joining us. But I'm going to ask this other question from Lisa. Body condition index is based off the eye patches. How transferable is that metric to other cetacean species that don't have an eye patch?
Good question. We're really lucky with the eye patches because it allows us a metric to longitudinally monitor the same individual across time. And the eye patch really shows us when the animals are level, if they, to reduce some of the biases there. But we've shown with the Antarctic killer whale work where, we didn't use the eye patches because they're so variable between types. We just selected a fixed distance behind the skull that showed that sensitive area. And I think we can do that with others. For example, work we do on gray whales and right whales, we identify the most variable measurement points. So on a right whale, it's 60% down the body where they keep these fat reserves caudally. It's part of their buoyancy issues too. And so you can identify these sensitive metric sites. And I think that's really important. When we're trying to look at change, we don't need to estimate mass or volume of the whale. We need sensitive measurement sites so we can look at change over time. And so I don't think it needs to be overly complicated. But no, I think that certainly for other odontocetes, that area behind the head is really important. And you could do it by a fixed distance between the blowhole and the dorsal fin, as we do for Antarctic killer whales. It's still a sensitive measure that changes over time. And the key thing is to try and do some power analysis, as we did, just to develop in methods that are the most sensitive. We've shown with southern residents, we started off measuring width at the anterior insertion of the dorsal fin, for example. Well, it's not very variable because there's a rib cage in the way. There's only so much change that can happen. But we know that behind the skull, there's a lot of scope for change because that's largely just fatty tissue. And so I think you can develop very similar metrics. We're just very lucky with the eye patch ratio that we've got some landmarks there that make it really easy and easy to do huge amounts of measuring quickly.
Nice. Thank you so much, John. Everyone is just saying thank you and nicely done, and we appreciate your time. So that's what's coming in on the chat right now. I'm hoping that we get you down here to Hatfield sometime in the relatively near future, and we continue this relationship and you continue to share with us. Just really enjoyed your work today. And I heard that you were talking about sharing an additional study with us in sometime in the future. So I'll hold you to that. I'll reach back out and say, all right, what's the next one? So thank you so much for your time. And for everybody online, thank you for joining us. It was definitely a great presentation. I'm happy to have you, and hopefully we'll see you again next week. And like I said, the recording for this will be out in hopefully a couple of weeks as those papers get in. Thank you, everybody. Lisa, anything to add?
No, thanks, except for doubling the thanks. Great talk, John. And I echo the sentiments of having you down in person as soon as we're all vaccinated.
I look forward to it. Thank you.
And until then, enjoy being in San Juan's and your time where you have to be at home. It's a good place to be stuck at home. I enjoy it up there a lot.
Yeah, I'm lucky. Well, thanks.
All right, everybody. Thanks so much. I'm going to end the presentation now, and hopefully we'll see you next week. Thank you very much, John. Appreciate it. Bye, Lisa.
Yeah.